Please, would you start your talk?
Okay, thank you. Well, first of all, I'd like to thank my hosts for having me here and being able to present some of the research that I've done throughout my career here. Now, second of all, while I'm an American holding a microphone, I am not going to perform karaoke up here, for which you should be very grateful. Now, I've had the pleasure in my career of working on a lot of farms, working with a lot of feedlots and dairies. And one of the things I've been preaching my entire life is everything is interconnected. And now this One Health approach has actually started to come to where it looks like I was ahead of the curve for a change, where we're all in this together. And that's the thing, the point that I try to make to my students and everybody we work with in research is that everything we do is interconnected with one another, and we are all in this same problem together. So with that, I'm going to try to take y'all on a little bit of a walk through the research that I've done, some of the data that we've encountered, and some of the solutions that we think we're on the verge of finding and utilizing, and things that can help you in the real world out in the field. Okay, so growth is not efficient. We all know this. Our cows are not efficient creatures. They're not little chickens. They are not big chickens or big pigs. They have a terrible efficiency compared to chickens and pigs, where it's about 7-to-1 versus the 1-to-3. But we know our lack of growth efficiency is due to things such as maintenance costs, non-growth dissipation, the microbial fermentation, which in itself is energetically inefficient. And the microbial maintenance cost varies. So ultimately, the cause of the low degree of efficiency of ruminants is because of the microbial population. So we'll come back to this gut microbes. But we can really improve, we can stand to improve dramatically this feed to gain ratio. Now the thing that we have to remember at all times is our feed costs are up to 75% of the costs of animal production, depending on what sector. So if we can improve this feed to gain even slightly, we can improve our producers' profitability because they're surviving on very narrow margins of profit. And that comes to why we actually feed antimicrobials in the first place. Because we really don't know why we feed them or why they work other than they work. We know that healthy animals are better at producing milk, meat, protein than animals that aren't sick. That's an easy thing to understand. And we know that if they're expending energy needlessly in combating these low-grade illnesses, that they're not going to grow as well. And again, best prevented illness is the best prevention. Prevention is the best cure, technically. Now, in the U.S., we've calculated that about 25% of all our antibiotics are used in growth-promoting functions. In total of all of our antibiotics, or 50 to 55% of antibiotics, are used in agriculture in one form or other. So we do play a big role in this antimicrobial resistance puzzle. Now, our subtherapeutic doses that are used for growth promotion are this 1/10 or often less, but they do improve growth performance, 5 to 20%. And if any of you in here can tell me why that happens, why feeding this low dose of antibiotics and result in such a significant improvement in animal performance, I'll definitely buy you a beer because I've been studying this my whole career and I can't get a good answer on this. I can't solve this puzzle. So any thoughts you have, I'm very welcome I would love to hear your input. Now, I'm going to tend to use pathogens and antimicrobial resistant organisms interchangeably throughout my talk. And that's because I've quit delineating them because there are so many of the foodborne pathogens that can carry antimicrobial resistance and so many antimicrobial resistant organisms that can be opportunistic pathogens. So I tend to lump them together interchangeably as these pathogen groups. But we live in countries that have a safe food supply, and it's growing safer every year. And that's a positive thing. It's something we do have to keep in mind. But in the U.S., our cost of foodborne illness is about $150 billion per year, caused primarily, in many cases, by bacteria. And the big ones that we have in the U.S. in our food supply, and it's similar in EU, both in scale and participants are Campylobacter, Salmonella, Escherichia coli, 0157H7 is the big player, the other E. coli, the enterohemorrhagic E. coli, and Listeria. All these guys count for about $40 billion a year in cost to the economy. Now, if you want to try to include antimicrobial resistance issues in there, this number can probably go up by a factor of 10, depending on how you want to lump these together. So antimicrobial resistance is a tremendous impactor economically. Now, there are the direct contamination issues. How do these pathogens get to our customers, to our consumers? Well, they can come from the animals, things like on the hides. As you pull a hide, you get a puff of dust off the hide that can be carrying pathogens, can be carrying your antimicrobial resistance organisms that are spread to the carcass. And as a cattleman, I love to show this slide of chicken, where we had fed chickens fluorescein dye immediately before they went into the processing plant. So when the crops ruptured, it spread this nice fluorescein dye on the carcass. So you could see it as an idea of where our pathogen spread actually occurs, where you can see it on this bird and on subsequent birds on the line after this one ruptured. So you can actually physically see that. So we love to show that at meetings where there's a lot of chicken people. They get very upset. But we can see the spread of pathogens, and again, this is not just a problem of cattle, but you can see pathogens spreading on the carcass, but they can also be spread intergenerationally, especially in the poultry industry. But for those of us with cattle interests, our calves, when they're right after they're born, they're licked by their dams, so you get this immediate vertical transmission throughout time between generations. Now, not only do you get your pathogens in the food supply, but you can get them in water. We've had several irrigation and runoff issues. Agricultural fair and open farms, where we have children coming from the large cities where they have never been exposed to animals. They come out and suddenly they're exposed to pathogens and antimicrobial resistant organisms, and the kids have lost the importance of hygiene. So they'll come out, pet the animals, let the goats lick their hands, and then they eat their sandwich without washing their hands in between. So we're seeing increasing problems with this. Issues from our confined animal feeding operations of dust blowing downwind is a significant problem. So we must account for these alternate routes. But there's one common factor that links all these, and that comes back to the gut. And as Hippocrates said, the gut is the center of all health. Okay, but beyond what Hippocrates could ever think of, the gut is far more complex than we ever imagined. The population is incredibly dense. We're talking 10 to the 10th cells per gram of feces. That's a tremendous number, and well over 15,000 different species of bacteria have been isolated at this point. We have protozoa, bacteria, fungi. So basically, if you can name an organism, you can pull it out of the intestinal tract of animals. And there's a lot of cross-feeding between our kingdoms, between protozoa, bacteria, archaea, and the host animal. So there's a lot of interlinks in the rumen and the gut of animals. It truly is an interlinked food web. Now, this gut microbiome that people have been talking about for about the past 7 to 10 years heavily is a lot more important than we realized. And some studies came out several years ago now, but they were very groundbreaking in the fact that germ-free mice could eat 30% more calories than their litter mates who were raised conventionally. So they're getting 1/3 more calories and they're still skinny, whereas the conventional mice were fat. So, I've always been very jealous of these mice being able to stay thin even though they eat more than everyone else. And interestingly, if you take the fecal transplant from these conventional mice to the germ-free mice, they quickly get fat. And that's because of the composition of their microbiome. And we don't really have time to get into that at this point, but it's really important in obesity. Now, microbial endocrinology refers to the fact that a lot of our microbes produce compounds, quorum-sensing molecules, that act like catecholamine hormones. So the bacteria actually release compounds that tell the body to perform a function, to release carbohydrate, for example, so that they can eat some energy, so that they can be supplied a nutrient they need. So the organisms are actually telling us what to do instead of vice versa as well. But we're starting to see more and more that the microbes are controlling us. Things like our mental health and autism. There's a role of microbes in autism as well as in depression. Now, ruminants, we know they're great for detoxification. There's a huge biochemical potential of enzymes in the rumen for the degradation of all kinds of feedstuffs and toxins. So it is truly a huge biochemical and genetic reservoir. But unfortunately, in this population, our antimicrobial resistance genes are easily maintained in this large crowd, this large commish of microbes. Okay, now when we start digging in a little more in depth into what the microbiome does, you know, obviously we understand that the volatile fatty acids are critical for production of animals, that we want to see acetate, propionate, and butyrate. For the feedlot cattle, we want to see a lot more propionate, but in all these cases, this is what this microbial fermentation drives the animal growth through the absorption. Now, it's important in gut health where we start seeing leaky gut syndrome if you're not getting enough VFA production. And it's also important for immunology and non-self recognition. Now, microbiologists have been telling us for years that diversity is extremely important, that we need to have a diverse microbial population. But one of the recent studies where they were looking at cattle who were efficient at producing milk, so they had a really good feed-to-gain ratio, that when you looked at what their microbial population was, the diversity was much lower than on cattle that were inefficient. Now, this goes against what everybody had always thought, that diversity was good for its own sake. But what they found was it was actually a specialized microbiota. In this case, very specialized for the high-grain diet and for being able to utilize grain very effectively and be able to degrade starch, that was the most efficient. So this kind of approach and understanding has changed a lot of our opinions and thoughts on what we need to do to improve animal production. Okay, so the microbes are always at war with one another, and their genes are ultimately the true unit of natural selection. But it's mediated by the microbial ecology and the microbial physiology. So the way the individual organisms are put together is what drives genetic selection. So the genes themselves are selected based on how the organisms are fit for their environment, whether it's the competition of nutrients, their affinity for nutrients, how fast they can grow, whether they're producing their own antimicrobial compounds. Because in some cases, organisms that produce their own antimicrobials are resistant to other types of antimicrobials in a similar function. So if you're making a weapon, you're already defended against your weapon. And that's something that we've really not looked into in depth as to what role that plays in the maintenance of antimicrobial resistance. Okay, now a lot of people have been talking about the core microbiome, that there's this population of microbes that never varies. In all cattle, you're going to have this. And that has been pushed as the most critical part of the population, that they are the most important players. And since the beginning of this concept, I've disagreed with that, and I still do. And my rationale for that goes back to a story by Sherlock Holmes, back in the 1800s. So this is a long time ago, a very famous detective in English history. And in one of the cases was solved because the dog in the house did not bark at whoever broke in, so that the dog knew the person that was coming in. So when we're looking at this core microbiome, is that the person we know, or is that the person we don't know? So are we looking at this the right way by focusing on this core microbiome? Personally, my belief is we need to look at the organisms that are different from one animal to another, not just this core microbiome that never varies. To me, those players are very unimportant in what we're wanting to see in improving food safety, animal production, and sustainability. All right, and one other kind of tidbit of linguistics that I'm a big believer in is people talk about antibiotic resistance. Well, not all bacteria are going to be sensitive to an antibiotic. Every antibiotic has its own mechanism of action. Some bacteria just lack the sensitivity. They lack the target for that antibiotic. So in the case of, say, a bacteriocin that punches a hole in the cell membrane, gram-negatives are going to be resistant to that naturally. They'll be insensitive to it, not truly what we would call resistant, where it's a developed thing. Now, bacteria that are sensitive, that do contain that target, can become resistant, and that's what our concern is. So there's a real difference here in these organisms, and people tend to lump insensitive and resistant together, and that's incorrect. But it's something you see a lot in the literature. But the resistance comes at a cost. So even if the organisms become resistant to an antibiotic, it does come at a cost to them. Even if you don't have the antibiotic around, there's a cost to maintaining these genes. And that allows us to start to target these organisms. Now, this was a study that I did a long time ago that illustrates this point really well. When you begin antimicrobial treatment, your resistant population goes up and your sensitive population goes down, which makes sense. When you remove the antibiotic treatment, that trend begins to reverse. So that shows there's an expense to maintaining this resistance as the organisms compete in that environment for nutrients. So going back to that previous slide about the microbial warfare, if you're expending an extra cost to maintain these genes in a vicious combat situation, you are going to have a reduction in your efficiency. So it does have a cost to maintain these genes. Additionally, if you have a major change in an antimicrobial resistance, if it is a significant change, say where the microbe has to put a glycocalyx outside the cell, that can also affect the efficiency of growth, even if your antibiotic isn't present. And it provides a competitive advantage. Now, we've all seen mobile DNA, and this is the wild card. This is the deck shuffling that bacteria do. And it can be done through plasmids or transposons or integrons. But the big one that we worry about primarily is plasmids. It's plasmid mediated. And basically a plasmid is just a small extracellular bit of DNA that you find in bacteria. And we heard a little more about them earlier, so I'm not going to go into depth on them. But basically, when bacteria connect to each other, they can pass this plasmid back and forth. And some strains of bacteria are extremely promiscuous, and they'll pass this back and forth with other organisms of the same species or same genus. Others will go across any genus. They will donate their plasmids to anything. So there's a lot of deck shuffling that can occur. But when you get these organisms that have the genes, if the antibiotic use does not sterilize the gut, does not sterilize the niche, then your growth is going to happen back from those same survivors, from that gene pool. And one of the interesting things that also happens a good bit is you start to see compensatory mutations in this population where they begin to change back because there is a cost to maintaining this antimicrobial resistance gene. So you can start to see organisms becoming sensitive again. Now, I get to go through the fun part of what have we looked at as ways to reduce these pathogens and antimicrobial resistant organisms. So collectively, all the approaches that we've looked at, and I'm going to touch on a good many of them, they can be clumped together as anti-pathogen strategies, those that look to give a competitive advantage to non-pathogenic, non-antimicrobial organisms, and some of the management factors. What can we do on the farm? So one of the great things that's come along in recent years is looking at bacteriophage. We've known about bacteriophage for just over 100 years now. And in several studies, and these are some of mine, bacteriophage are just viruses that kill bacteria. That's all they are. It's basically giving a fatal cold to a bacterium where it hijacks their cellular membranes and takes over all their processes. But you can see a difference in these pathogens where phage reduce the pathogen presence in the gut. And this was in pigs. This was done in cattle as well. Now, one of the interesting things, and I've got a bad habit of telling people about my failures as a scientist, but in this study, we had predicted that the best dose where we would see the greatest reduction in pathogens would be at this one, because if you put a little bit of phage in, it's good. If you put more in, it's going to be better. Because I grew up on a farm. That was my thought, so I was trying to see how high we could put this up and kill it. But what we found is when we increased our phage level, it actually was counterproductive because the phage began to interfere with each other when they were trying to take over the bacterial cells. So this was one of those great moments where biology tells you you're not quite as smart as you think you are. So our lower levels were the most effective. So this is just an example of the approach of bacteriophage, that we can use them because not only do some bacteriophage carry genes back and forth, but some are strictly lytic, where they come in and they rupture these bacterial cells and can kill them. So we can use this and harness them to go after specific organisms. Now, sodium chlorate is another product that we played with for several years. And all it is it's a sodium salt of chlorate. And bacteria that are equipped with nitrate reductase, such as most Enterobacteriaceae, most gut bugs are equipped with many gut bugs, not most, are equipped with nitrate reductase. And what it does is it takes nitrate to nitrite inside the bacterial cell. No big deal, but what nitrate reductase does is it takes chlorate to chlorite. It does this just the same way it's doing nitrate. Well, chlorite will kill the bacterial cell. So that wipes out your organism. So that's a great thing. So if we feed that to cattle, it reduces pathogens in the animal. And we did this with a study where I'd infected cattle with 3 different strains of E. coli 0157H7 that were resistant to 3 different antibiotics. So we had the AMR spattered in there as well. And what we saw was a reduction in total coliforms and E. coli, as well as all strains of the fatal 0157H7. So we had significant reduction simply by putting sodium chlorate into the water of these animals, letting them drink it for 24 hours before we process them, before we simulated their transport and process them. So this way it's something we can do on the plant or right before the animals go to the plant to be able to reduce pathogens while they're going through that stressful period of transition into a processing facility. Now, we've used antibiotics for years as a way to improve animal health. And they still have a role, especially those that are not used in human health. And as long as we have responsible use and have proper selection, we can still balance that risk and reward equation properly. Now, one of the products that we've looked at for a few years, and this was done by Dr. Boulder years ago, where flavomycin, or phospholipol, it's hard for me to say that word still, but it was able to reduce the populations of Salmonella enteritidis in chickens. Furthermore, other studies have shown that flavomycin could reduce the percentage of pigs that were shedding Salmonella. And other studies have shown a positive impact of feeding on growth. So, okay, this is great. We've got a lot of antibiotics that do this. But where the twist in this story comes from is curing of plasmids. And by curing of plasmids, it just means removing plasmids from the population. And this is a story that has utterly fascinated me for about 3 years now, and I've been trying to get to the basis of it and try to get to the understanding of it. And I'll cut to the end of that story and tell you, I don't know the answer to this one. I haven't been able to get the perfect experimental setup. But what has been shown is when you look at the degree of antimicrobial resistance, and in this case they were looking at AMP and oxy, animals that had no antibiotics had a pretty high level of resistance. Those that were treated with avaparsin had a pretty high degree of resistance. But the ones that were fed flavomycin had a very low, it was almost non-existent, which was an intriguing discovery. That's very strange, and this goes with other studies in vitro where they've seen this reduction in plasmid carriage in these organisms. Now, this is another study that was done in chickens in this case, but it also shows again that the flavomycin treatment over time had reduced the incidence and the prevalence of these antimicrobial resistant organisms. And in this study, they looked at ampicillin-resistant ceftiofur, ceftriaxone, a whole variety of antibiotics. And it was all seen that it functioned this way, that it reduced these pathogens. So to me, this is a fascinating story. Why does this compound do this? Why does it have this antimicrobial action against antiplasmid carrying capacity as well as an antimicrobial property? How does that work? Okay, other approaches. So now we've gone through some of the pathogen reducing ones. Now let's look at a couple of ideas of of competitive enhancement. How do we use competitive exclusion or probiotics or prebiotics to improve food safety? Now in the US, our direct fed microbials are what most people call probiotics. So that's the technical term in the US. But lumped together, it includes probiotics, which are the live organisms, whether it's bacterial or fungal or end products, so there's fermentations. Whether it's prebiotics, such as oligosaccharides of different forms, or competitive exclusion cultures, where you're using a live culture from an animal, from an adult animal, and putting it in a neonate to be able to exclude pathogens from their environment. So, overall, do these probiotics and prebiotics work? Yes, they do at times, but in some ways, it's a form of magic because it's dependent on the organism that you're using, and what the environment is, what the selective pressures in that environment are. And for example, we can follow, in this case, we were looking at antimicrobial resistant E. coli and Salmonella typhimurium, where we fed a yeast product. In this case, this was an in vitro study, so it was a lot simpler than actually feeding it. But we were including it, and you could see a dramatic decrease in the MDR Salmonella typhimurium that we were using in this study. And the E. coli populations, even as we increased the concentrations, the E. coli remained fairly constant, but there was a significant drop in our Salmonella typhimurium that was MDR. But these direct-fed microbials or probiotics have the capacity to stimulate this microbial flora. Again, remember that power of the microbial flora to affect the entire host, this holistic approach, that if we can add this, we can stimulate the native flora through either these unidentified growth factors or by providing a new member to that microbial population who may benefit. Now, one of the problems we run into in the industry is what we call magic foo-foo dust. We have a lot of companies that will tell you, my probiotic will make your cows pretty, their hair will be shiny, they will drive the tractor for you, they'll come in your house and wash your dishes, they're going to do everything, and they'll grow better. So you have people telling you these great stories. And the big thing to look at with these companies, and as veterinarians, you'll have these nutritionists and these salespeople that will tell you all these great stories on their probiotics. And one of the big tricks that I've picked up over the years is if you ask a salesperson, what's the organism in your product? Most of the times they're going to say, well, it's a lactic acid bacteria. They won't know the name of the organism. And that's critical. That's one of the big things to understanding how these organisms work, is knowing who is there. But you'd be surprised at how many companies do not know what organisms are in their products. Now, or at least their salespeople don't know. I'm sure the companies do. And this is where metagenomics and next-generation sequencing opens a lot of doors to understanding. Now, diet also can change the microbial population. This is getting into management strategies and how can the management be utilized to help reduce these pathogens. So we can add a change in the diet, going from a high grain to a hay ration, and see significant reduction in E. coli shedding. And if we look at some other dietary components, such as essential oils, we can see a reduction in pathogens and an increase in growth performance. Citrus pulp and peel is a great byproduct that we use a lot in the U.S. in cattle, and it can reduce Salmonella populations as well as E. coli 0157H7, and it doesn't affect Campylobacter populations, but it improves animal growth. So it's a really exciting product because it contains these D-limonene and a few other essential oils, but it's a byproduct of juice making so that we can actually get this cheap for producers to be able to feed to animals. Now, stress is also one of those things that the animals experience a lot in their lifetime. And when you see them being moved from place to place or being handled, run through a chute, you know they're going through stress where their norepinephrine increases. Well, bacteria such as Salmonella actually feed on norepinephrine, going back to what I'd said earlier about the quorum-sensing molecules and their likeness to hormones. Well, when norepinephrine is injected into a culture of Salmonella, your populations will increase dramatically. When you look at Salmonella concentrations in pigs, in this case, as they were controlled, mixed with pigs from other pens that they'd never met before, transported mixing, you can see an increase in the Salmonella concentration. In this case, we were just looking at ileal contents. And others have looked at on-farm right before transport when they've grouped the pigs together, after they're transported and after layerage, and you can see a significant increase in microbial populations of pathogens following these kind of stresses. So just simply stresses themselves, in addition, are even separate from mixing with new pathogens they've never been exposed to, just the mixing stresses alone can be significant. And this was a study that I did a long time ago now, farther back than I realized until I just looked at this, but where we took some pigs, newborn pigs, or newly weaned pigs, and we would mix them back and forth from one pen to the other every day where they had to reestablish their dominant structures. So this was a form of social stresses and social mixing where the animals had to interact every day. And you can see a decrease in the, or an increase, in the mixed groups, the yellow bars, caused by just this simple social stress. And later studies showed that social stresses can cause a significant change in the microbial population makeup of the gut. So that stresses alone, again, this comes back to the idea of this two-way communication between the host animal and the microbes, that the stresses that the animal experiences affect this microbial population. And how can we utilize that and those kind of impactors on our, to be able to reduce these pathogens in our animals. Now finally, when you get to the back end of the animal and animal production, we have to get rid of the manure. And manure is always a problem, but you can take it in the U.S. and either irrigate it in a liquid form, slurry inject it, or apply it to your soil surface. But all these have certain problems with them, especially when it comes to looking at the presence of antimicrobial resistance genes, because if you're hiding these genes in the soil, sooner or later they're going to come back. And these great new ways of mitigating antimicrobial resistance genes comes in with wood chip bioreactors where you can get 100% or 70 to 80% reduction, composting, or just straight land application. where over several months to a year time, you can reduce those populations of the genes, but it takes a long time. Anaerobic digestion works, but the really exciting one is thermal processing. If you can heat your manure up, which is some of what is occurring in composting, if you can heat your manure up enough, you can get up to 100% removal, not only of the genes, but of the bacteria as well, which is a really exciting development in being able to handle manure in large scales. So with that, I wanted to touch on the whole variety of things that we face in the animal industry. But we've got to remember, antibiotics have done wonders. But the sensitivity to antibiotics, it's this natural resource, and we've squandered a lot of that through indiscriminate use of antibiotics. But it may be renewable. Things like flavomycin treatment or stresses in specific ways to increase the resistance again, to bring that back, may be useful to be able to harness that power of this microbiome, this vast biochemical reservoir that we can actually reach into and pull some of the genes out of. But ultimately, we're going to have to use a combination of anti-pathogen, pro-competition, and good animal management, and good animal health. And this comes back all together to this One Health process, that we are all in this together, and there's not one section that can look at the other and say, well, you guys caused this problem, you guys fix it. We're all tied together because everything is connected on this. Unfortunately, we don't have a silver bullet. I can't stand up here and tell you, here's how we reduce antimicrobial resistance in animals. I wish I could. But I've given you a taste of some of the methods that have been researched by myself and other groups around the world in the past 15 years of ways to be able to actually go on farm and reduce these pathogens. But whatever we do, whatever approach we take, we've got to remember that we want our producers, whatever we put in place, they need to be able to take what we're putting on the farm and make more animal, whether it's more milk or more animal out of that. We need to provide them a profit margin in these approaches, or it has to be regulated for them to be able to do this. Otherwise, their good farmers who put these into place are going to be put out of business by market forces. So we really have to make these improvements pay for themselves. And that's a real challenge. So with that, I'd like to thank you for your time, and again, thank the organizers for hosting me. And if you have any questions, I'll be happy to try to answer them. And thank y'all.
So thank you very much for